Now, gene linkage occurs when genes are close together on a single chromosome. So when you have genes, and obviously, since remembering, I'm going to say it again, that a chromosome is a double helix. A chromosome is this double helix idea with a whole sequence of DNA, which is again a polymer of nucleic acids, deoxyribonucleic acids, all in a row. And only parts of this are actual genes. So when we look at this might be one gene, and there might be some more regulatory space, and then there's another gene, and then there's some more regulatory space, and then there's another gene, and then some more regulatory space, and then even another gene, and it continues. And the genes, as I say, there'll be all kinds of sequences that are going to give you all kinds of proteins throughout the entire genome. So when we're looking at a gene, you're looking at only portions of the DNA that code for an mRNA. And most of the time, or a lot of the time, a protein. Remember that some genes don't code for proteins, they only code for RNAs. So these genes that are transcribed and then translated are found throughout the chromosome. In this drawing here, you only have these lines, and we're only looking, and in your activity you have popsicle sticks. But always remember, we're talking about this double helix, this polymer of DNA. And of course, each gene, each chromosome, you have two chromosomes, one from mom and one from dad. You got one of your chromosomes from your mother, and you got one of your chromosomes from your father. And when you have a homologous chromosome, you're talking about a chromosome that has the same genes. So the W genes are both found here, and the F genes are both found here, and the N genes are both found here. If these genes, right, and then of course the H gene would be found on yet another chromosome, so there's another chromosome that has a pair. Remember, they're homologous pairs. Every chromosome has its twin, except for X and Y, which they're the pairs that you get, the sex chromosomes you get. In the female, they are twins, they're two X's, but in the male, there's an X and a Y. And you get every chromosome you have, every pair of chromosomes, every homologous pair, you get one from mom and the other one from dad. That's what you inherited. Those are the particles that Mendel described. He didn't know what they were at the time, but he described them as particles. And this would be then here along on this chromosome, you'd find the H. So H's would be here, and big H is bighorn, and little h is little horn. And then, with the yellow, with the W, you have wings, big W is wings. And little w is no wings. As I said, I don't like using these letters because it's sometimes difficult to tell whether you're dealing with a big W or a little w unless you're typing it. And even then, it could be somewhat confusing. And then the F would be fire breathing. And little f, no fire. And N would be long fangs. N would be long fangs. and small n short. Remember that these would be the phenotypes. These would be the phenotypes. The symbol N or WF, those are the genes. The phenotype would be what you get at the end. The phenotype depends on the combination of genes. Okay, so here it shows you have a homologous pair, and the three genes are close together on the same chromosome. Now you might have another gene down here that is, Z, and it codes for whatever. This gene is far away from the other chromosomes, from the other genes. This gene is pretty far away. So whenever crossing over happens, these can independently assort, because if crossing over happens, remember what crossing over is. Let me review with you what crossing over is. You have a chromosome from mom that's been copied. So these are sister chromatids. And these sister chromatids would have the same alleles. So if mom has big A, then both are big A. And then you have the other homologous chromosome, the pair, from dad, that you got from dad. When you're ready to replicate, you make copies through the during S phase of the cell cycle. And that was copied as well, but maybe dad has a different allele, so little a, little a. Now, during crossing over, remember that this is going to form a tetrad. So when you're looking during prophase 1 of meiosis, these two form a tetrad. They come together, and they align together. When they get close enough, this portion of the DNA on this sister chromatid here, and on this sister chromatid, this whole portion of the DNA switch. They break and they exchange that DNA. So what you get is one sister chromatid that has little a, another sister chromatid then that has part of mom's, and that part of mom's has a big A. The rest is still dad's. Then the other homolog has now part of dad's chromosome, which has that little a, and part of mom's chromosome, which is that has all the rest of that gene, all the rest of the genes that are on that chromosome, whatever those are. And then here you have a big A. Again, all held together by a centromere. So when you're looking at these chromosomes, when the genes are far apart, the way Z in this example is far apart from N, the W, and the F, notice they're very far apart compared to what these three together. They're not going to necessarily travel together because crossing over could happen. And if they cross over, then big A is not always going to be found with whatever the other three are. So if the crossing over happens here in between these two sections of the DNA, then these three won't always travel with big Z, and these three won't always travel with little z, because they could switch, at least on one of the two sister chromatids, and when they switch, then they're no longer traveling together, so they're no longer linked. So the linkage, there's different percentage linkage, so you can get linkage to the closer the genes are on the chromosome, the more linked they are. So you have to think of linkage linkage is all about how close are these genes to each other. And crossing over during prophase 1 of meiosis means that you're going to mix up some of the DNA, especially if the genes are far apart from each other. So this purple gene is not necessarily very well linked to this yellow gene, but the yellow, the blue, and the pink are very, very closely linked together. So you have to consider, when you're looking at genes, how close are they together when you're considering linkage. So when we say is there genetic linkage, they have to be on the same chromosome. They also have to be close together. For the sake of this project, you're going to go ahead and assume that they're close together and that they're very much like this colored example here, this color-coded example you have here. So that W is the pink is the yellow. So you have let me see if I can, it won't work because I have, all right, there you go. So yellow is the W, so you have big W, you have little w. The F is blue, so you have big F, and you have little f. And then N is the pink, so big N and little n. Now, again, you got one of these chromosomes from mom and the other one from dad. Or this parent got one from mom and one from dad, right? This father got one of these from their father and one from their mother. This mother got one from their father and one from their mother. It so happens that the mother and father of the mother's mother and father, the grandparents of the child, they gave the mother both. They both gave their mother the homozygous recessive. So each of these three genes, they gave the recessive form of those genes, right? The genes, I should say, the protein that was produced by these genes leads to recessive phenotype, right? So these genes, because they're lowercase, and they produce a recessive phenotype. The father has the dominant phenotype gene and the recessive phenotype gene, and so one of the father's parents gave him all three recessive genes on one chromosome, and all three of the dominant forms of the dominant phenotype genes on the other chromosome. So he's heterozygous for all three genes: big W, big F, big N, little n. And the mom is little w, little f, and little n. So the genotype of the father is heterozygous for all three, and the mother's homozygous for all three, homozygous recessive. The other interesting thing is that they're very close together, so these are linked. They're not going to independently assort. Again, this is where Mendel got lucky, because the genes he studied were far apart. Number one, they only coded for the one trait, so whatever they coded for that impacted that trait directly. There were no other genes involved with that trait, and secondly, they weren't linked with the other genes the other gene products he was looking at. So the phenotypes were all dependent on one single gene, and those genes were not linked to each other, so they independently assorted. That allowed him to come up with his law of independent assortment, which is, again, a law is what he sees, what he saw. He explained it with the model, with the theory that these are particles that get passed on from one person to the next, from one generation to the next. And today we call those chromosomes. Now, you draw the genes for the chromosome, two pairs for the father and the mother. And of course, as we said, the chromosome on the mother and father, there's only H to consider. So this was the genotype for the father, and let's go ahead and highlight that. So the father's heterozygous for all the genes, and the mother's is homozygous recessive for all of the genes. So again, for the phenotype is recessive, and it's homozygous. So when we look at the mom, she has a chromosome that has an H, a little h. She has a chromosome here with a little h. And here the father has one chromosome that has a big H and one with a little h. Now, to be sure, this other chromosome has other genes, but we're not looking at those genes. We're not looking at the gene products. We're not looking at those phenotypes. We're only looking at whether they have wings, or do they breathe fire, do they have short or long fangs, do they have big or short horns, or big or small horns. And so what we find is that these three dominant or recessive phenotypes and the genes that produce them are found close together. The genes are found close together on one chromosome. So they are linked. They're going to move as a group. So you're either going to have big W, big F, big N, or little w, little f, little n. They're not gonna separate. You can't mix them up. They're only going to be able to produce. When you look at this, these three genes are going to move together, and these three alleles are going to be moving together. So when you look at the father with the H, now the H is not the horns, big horns or small horns, is not linked. In other words, it's on a different chromosome. So remember, you can see it right here. It says chromosome number 2 versus chromosome number 1. So these are two different sets of chromosomes. So here's where some of the people that are very artistic could take hold, but you'd have a dragon with, in this case, long fangs, because father has a big N, so it's going to produce long fangs. It's going to breathe fire. It's going to have wings. It's going to I would just wish I was a better artist. I know there's a bunch of you out there that are much better artists than me, and I'm often very, I wouldn't say jealous, but I do have some envy for you. And so in this case, he has a big horn because he has one big H, so he has a big horn. And he does have wings. Now the mom is going to have, again, she is not going to, you know what, maybe I can save some time and duplicate this. So by duplicating it, I know that mom has little horns, so the horns are going to be smaller than the dad's. Be smaller. And they're not going to be fire breathers because they have both the forms of the genes that lead to the recessive phenotype, and that would be no fire breathing. And it has this mother has no wings in this case. I'm sure some of you have drawn much better dragons than I'm drawing here. All right. And the fangs are short. So I should... So where the dad has these big, long fangs, the mother has a very... So as you can see, that there's a very significant difference in the phenotype between the mother and the father because of these genes that they inherited from their mother and father. Remembering wherever you see the dominant form, the gene that produces the dominant form of the phenotype, we call it we capitalize that symbol that represents that gene. And remember, each symbol then represents a series of nucleic acids, right? And that there's a difference between these two sequences of nucleic acids. It's small. They still produce. They still impact the same phenotype, but I mean it might be a large difference. But in any case, the difference between they still are the same gene. They're in the same location. They're in the same place on the chromosome. It's just that instead of an A, you have a T. Instead of a G, you have a C, or whatever the difference is between the two sequences. And that's true of all alleles. These two are the exact same sequences. These two are the exact same sequences. And these two are the exact same sequences. And they lead to the same phenotype. In this case, small horns, no fire breathing, and no wings. And they small horns. I'm sorry. This is small fangs. And so you have small fangs, short fangs. Here you have long fangs, large horns. They do have wings, and they have, and they're for breathing fire. Okay. Now, when you're doing this multiple alleles, and you could start thinking about this, and this is going to be, it can be very complicated. In other words, you could try to figure out instead of doing a FOIL. Remember, we did big H, little h, big W, little w, and big N, little n, or these two right here. We would do FOIL, and we'd see this could produce a gamete that has big H, big W. It could produce a gamete that has big H, little w. And we've done this before several times. Big H, little w, and it can produce a gamete that can produce little h, big W, and little h, little w. So there's 4 different gametes, 4 different sperm, or 4 different eggs that it could produce, and we've talked about that. Now, what's interesting here is and this is very, very key because it's going to save you a lot of work. When you look at this particular set of genes, this big W, big F, big N, little w, little f, little n, they're going to move together. They're going to move together. So because they're linked, if big W, big F, big N move over here, and little w, little f move over here, you got big H here, little h there, then you're going to have 2 separate possible gene combinations. Now notice if they weren't linked, then you would have a very complex series of more complex than we have here, series of considerations, because then big W, big F, big N, little n, and big H, little h that were not linked, then you could get big W, big F, big N, and big H. So you have big W, big F, big N, big H, or you could get big W, little f, etc. You'd have all kinds of various possible combinations. But because those three are linked, then you're only dealing with one unit for all intents and purposes. These 3 are going to move together. So that's really important, because that allows us now to look at this and consider only where these 3 are going, not where each individual might go and what different combinations you're going to have. And of course, you're going to have this moving together, this one moving separately, this moving separately, this moving separately. So you do have to consider that option, that variation. So in this one, since you're going to get, let's go ahead and just do it. Remembering again, as I said earlier, when you're dealing with, and this is also big W, big F, big N, right? And we're not considering crossing over, but even if we considered crossing over, these 3 are going to move together, and these 3 are going to move together. They're not going to cross over in between because they're linked. That's what linked means. There's not going to be any crossing over in between them. They're very close together on the chromosome, where here they can cross over, but they're going to move. A big H is going to move, and little h is going to move into separate chromosomes. in separate cells, no matter what. All right. So here's big W, big F, big. Let me make it smaller here so we can read it. It's difficult to read when it's not being neat. So big W, big F, big N. This is obviously big H. So big W, big F, big N is this one, and this is big H, and this is big W, big F, big N, and this is big H. So here you have big W, big F, big N, big H. On this one, you have little w, little f. Again, all of them are moving together. And little h, little w, little f, little n, little h. Little w, little f, little n, little h. So now you have your combination. So there's only really, if little h goes here and big H goes here, so all the dominant phenotype producing genes come together, and all the recessive phenotype producing genes come up in the other cell. Then you're going to have these two gametes that are possible. Probability, possible. Now, if on the other hand this H, instead of going right and that big H going left, if it were reversed and big H went with the other unit of 3 genes, you'd get this, right? And remember that they're copies on each cell. I'm just going to go ahead and just do it on this one version rather than writing them out here. But you know what? I'm going to go ahead and do it. It's big W, big F, and big N are here, and little h is here, because remember, they're copies. These are sister chromatids. My hope is that by keep repeating it over and over and over again, the vocabulary, and I hope you're doing the same, that it's going to become more solidified in your mind what we're talking about here. Those sister chromatids are identical, and these three are going to move as a unit. They're moving as a unit, and these 3 are moving as a unit, and these 3 are moving as a unit, and these 3 are moving as a unit. And of course, this is moving, and this is moving, and this is moving, and this is moving separately as well. These are independently assorting, but these 3 genes don't independently assort because they're on the same chromosome and they're close together. So these 3 don't independently assort, but these 3 versus this one, these 2 do independently assort. That's why we have these 4 possible gametes. So here you have big W, big F, big N, which is what we would get here, big W, big F, big N. And here you'd have a little h. Here you'd have big W, big F, big N, and you'd have a little h. So you have big W, big F, big N, little h. Or you can have little w, little f, little n, big H, little w, little f, little n, big H. Little w, little f, little n, big H. So these are 4 different phenotypes. It's the exact same 4. And previously, I would have been very worried that we'd have to deal with 4 different possible combination genes and 16 different possible gametes, all kinds of strange combinations. But no, these are the only 4 possible gametes because these 3 genes are linked. You're limited to the phenotypes that you could possibly get. So it asks the question, how many different combinations of alleles can be found in the eggs produced by the homozygous mother dragon? Homozygous mother dragon. How many different combinations of alleles? Well, that seems pretty straightforward. There's only one, right? She can only produce a little w, little f, little n, little h egg. That's all she can do. She'll produce 4 of them, but that's all they're all going to be little w, little f, little n, little h. Why? Because that's all she has to give. There they are. She might get different ones, but they're all going to be little w, little f, little n, little h. all 4 of her eggs, little w, little f, little n, little h. Now the father's genes, though, the father can produce all 4 of these possible gametes. Because he's heterozygous, he can produce all 4 of these, but the mother can only produce one possible outcome. So, because these are going to move, because these can move as a unit, because these are the gametes that they can produce, we can make a Punnett Square, adapt it a little bit. Might look a little weird to you, but mom can give little w, little f, little n, little h. We've already identified the different types of gametes that dad can make. So mom can only make this. The mother can only make this gamete, this one type of egg. The father can make these 4 types of sperm. He can make big W, big F, big N, big H. Or he could make the other one, and here it is, little w, little f, little n, little h. Or he could make a sperm that has big W, big F, big N, little h. Or he could make, or he can make little w, little f, little n, big H. So now, when you combine these two, you can get, and you know what, I'm going to color code these. And I'm going to say little w, little f, little n, little h coming from mom. And I'm going to say little w, big F, big N, big H coming from dad. It's a little genderist, but I'm going to go ahead and do this just to be clear. Hopefully the color coding helps. So what we would get is little w, we'd get little f from mom and big F from dad, and we'd get little n from mom and big N from dad. You know what? I'm going to have to erase this and zoom in a little bit because it's going to be hard to see. It's going to be little w, little f, little n, and little h. And it's going to be, dad can give a little w, big F, big N, big H. And again, mom is only going to give these lowercase versions of the genes that can produce the recessive phenotype, and dad can give either one. And that genetic variation is going to lead to 4 different kinds of gametes, 4 different zygotes. And those 4 zygotes are going to lead to 4 different brothers and sisters. And that explains why brothers and sisters can look so different. They can look so different because and yet still be similar because they can share certain traits and not others. They can share certain genes and not others. So that's what the Punnett Square would look like. Now you could, if you really wanted to, you could go ahead and put 4 more, the rest of these rows, and up for a Punnett Square and do a 4 by, and then end up with 16. But these 3 would be the same as the first one. So you just would be repeating yourself. There's no need to do that. Mathematically, 1 out of 4 is the same as 4 out of 16, right? They're equal to each other. They can be reduced to equal to each other. So we don't need to concern ourselves, right, with it just jammed again. Just jammed. So doing the Punnett Square would look like this, and you could have done this here. But I think by color coding it, I hope you see where I'm going with this. I hope by color coding you can see where I'm going with this, that the Father gave these possible genes that we figured out here by remembering, and what made our life easier is recognizing that these 3 move together. That these 3, once they form what they form, they are going to move together. So they're going to be little w, little f, little n, right? Or it's going to be big W, big F, big N. So those 3 are going to move together. Now again, we're not worrying about crossing over, because even if there was crossing over, there wouldn't be any crossing over in between these 3. So you can still treat these 3 as a unit. So any genes that are linked can be treated as a unit when you're trying to figure out probabilities. Now here is a problem, and this is a problem that happens every time I use W's. This is a big W, big F, big N. Remember, that came from here. That was big W, big F, big N, and a big H. And this is big W, big F, big N, and a capital H. So this actually should be a big W, and that was a mistake I made because I wasn't careful when I transcribed the letters. So this is a capital letter, W. So this is going to produce not the lowercase version, not the version of the gene that produces the recessive allele, but the uppercase version, the form of the gene that produces the dominant phenotype. So this is important when you're trying to figure out whether something has wings or not. So yes, these 2 will have wings. The others don't. And in fact, we can say that with some assurity that this is going to have fire breathing, and this one's going to have fire breathing. And that fire would come from here, and that fire would come from here. All right, and then big-horned, you can say, sure, this is going to have big horn. And this one's going to have big horn, the dominant phenotype. And breathing fire, fangs. This is going to produce long fangs. And this one's going to be long fangs. And those are your dominant phenotypes. So when we're looking at, and the question they ask you here is, which one will there be any? The answer is yes. But it says there'll be 2 that have wings, but the question is, will there any dominant allele for wings, but not dominant for fire breathing? Well, wings, this has wings and fire, and this has wings and fire, so no, the answer is no. There are none that would have wings, but not have fire breathing. Explain the difference in results in inheritance of wing and fire breathing genes versus the inheritance of wing and horn. So wing and fire breathing, and this goes back to the whole linkage, right? Big W, big F, big N are going to be together. So anyone that is fire breathing, has wings, fire breathing, I'm sorry, that's not true. Big wings, fire-breathing, and long fangs are going to be together. These three are going to be together. Why are these three together? Why are these three together? They're always together because they're linked. These three genes are linked. So why is it that you're never going to have fire breathing wings without fire breathing? Because they're always going to be moving together. Now, big horns move separately. They're on a separate chromosome. So you can get a big horn with no wings, no fire breathing, and short fangs. You can get a big horn, or you can get a little horn with wings, fire breathing, and long fangs. Or you can get a big horn with all three. So big horn can have all kinds of variety because they're on a completely separate chromosome. But these three are linked, so they're going to move together. Wherever you have wings, they have fire and long fangs. You have long fangs, you have fire and wings. Why? Because they're linked. That explains that. So the genes are located near each other on the same chromosome will move together during meiosis and fertilization. Therefore, inheritance of these genes is linked, as I just said. It is referred to as genetic linkage. As discussed previously, genes on different chromosomes are inherited independently, independent assortment, in accordance with the law of independent assortment. Remember, the law of independent assortment is just that they can be found in a random distribution in different cells because they're on different chromosomes and they assort during meiosis separately. Even if they're on the same chromosome, if they're far apart, because of crossing over, they can independently assort or be more independently assorting. How's that, if that helps you a little better? In the chart below, indicate 4 possible genotypes of the baby dragon produced and by this mother and father draw the phenotype and genotype. Well, I'm not going to draw the phenotype on each of those, but you can see that for each of these you can draw one that has wings, fire, big horns, long fangs, wings, fire, long fangs, no wings, no fire, short fangs, and short horns, no wings, no fire, short fangs, and big horn. So you can draw those 4 phenotypes. Will any of the baby dragons with wings have short fangs? Wings with short fangs? No. Why? Because those genes are linked. Will any of the fire-breathing dragons have short fangs? No. Why? Because they're linked. Right? Again, if they have fire breathing, they have long fangs. They have wings and they have long fangs. So anybody with wings has long fangs and fire breathing. So those 3 are linked together. Sorry, it's getting late and my mind is drifting. Here's will any of the fire breathing baby dragons have no wings? Fire breathing baby dragons have no wings. No. Why? Because they're linked. I don't know what else to say about that. Remember, it's fire breathing is big F, wings is big W, and long fangs is big N. Those 3 move together. So again, because the genes move together, if you have one, you have the other. They're too close together on the same chromosome, so they're going to move together as they're separated through meiosis. As homologous chromosomes in prophase 1, they come together and form a tetrad. Even if there's crossing over, it's not going to happen between these 3 because they're too close together. Then then the 2 homologs are going to get separated in meiosis 1. Then the 2 sister chromatids separated in meiosis 2. But because these 3 are moving together, anytime you have fire breathing, you have wings and you have a long fang. Based on your drawings in the above chart and the Punnett Square on the previous page, answer the following 2 questions. On average, what fraction of baby dragons with big horns will be fire breathers. So again, I'm not going to go through the drawings, but if you take a look, it says on average. They say on average because remember, any given set of babies, you can have a repeat. So you can, if these two parents had baby dragons, they could have 100 dragons. They could all have wings, fire-breathing, big horns, and long fangs. All 100 babies could have that. But if they had 1,000,000 babies, then it would come closer and closer to 1/4 each, right? So again, the larger your sample size, the closer you get to that predicted value, just because of randomness. That's what randomness means. So on average, what fraction of the baby dragons with big horns will be fire breathers? What fraction of the baby dragons with big horns be fire? So here's big horn, here's big horn. There's 2 with big horns, only 1 has fire breathing, so it's 1 out of 2. 1 out of 2. On average, what fraction of baby dragons with small horns will be fire breathing? Well, small horn, small horn. This is small horn, and I should write it in. What fraction? This is also small horn. So this is small horn, no fire breathing, short fang, no wings. This is small horn, wings, fire breathing, long fangs. This is big horn, this is big horn. So 2 have small horns. How many have fire breathing? Only 1. So again, 1/2. So why is it that we only have 1/2 that have small horn and fire breathing? Well, the small horn independently assorts because it's on a separate chromosome. And because small horns can independently assort, you can get either, and the parent is heterozygous, you'll get small horn happening in 2 of the 4 possible gametes that it can produce. So you see, there's little h and there's little h. The other 2 are going to have the big H's. It's just a simple idea that you're going to have independent assortment. Now, the 3 genes that are linked will move together, but when you consider that group of 3 as compared to the horn gene, and the group of 3, the linked genes, will independently assort. So because of that, you can have 4 different combinations of genes. The mother can only produce one type of gamete. All 4 of the genes are going to produce the recessive phenotype. And so what you get is only 2 of them having small horns. And of those 2, only 1 of them, or 1/2 of them, is going to have fire. Is going to be fire breathing. So that kind of hopefully that helps you explain the law of independent assortment and the principles of genetics, and to explain why only half of each of these, big horn and small horns, are going to have fire breathing. So now they ask you that in the principles of inheritance in these dragons also apply to inheritance in humans, other animals, and plants. However, inheritance in humans, other animals, and plants is much more complex than inheritance in these particular dragons. And we alluded to some of that whole crossing over business. There's also a much more complex layer of genetic interaction, but let's just talk about that, because right now we're talking about dominance and recessive, but there are other types of impacts due to difference in transcription and translation. But we'll talk about that and how genes interact. So first, these 2 principles of inheritance illustrated by first, list two principles of inheritance illustrated by the dragon genetics activity. Now, I've said them over and over and over again in both these videos that I'm posting tonight. So you should be able to write those in yourself. I'm not going to tell you the answer to those. What are some additional complexity of inheritance in humans, other animals, and plants? Discuss the following questions. Genetic linkage is very strong for genes which are located close to each other on the same chromosome. What happens in the case of two genes which are far apart in the same chromosome? We talked about that. That was crossing over. Are all alleles either completely dominant or completely recessive? The answer is no, and we're going to talk about these complexities in class, and that's going to be the nature of our discussion in the second half of this week. So hopefully you get this, you're able to review it, and you get these answers written out. Those of you that have class tomorrow and are going to get this late, I'll run a portion of this in class to let you look at it, but I'll also post it on your Schoology account. All right, guys, I hope this helped.